Hello everyone, welcome to another video of our channel. Today in this video we will discuss about the chromosome, their constituents, their structure, their functions and other details. So now let's start. Now at first we will have to know what does the word chromosome means. The word chromosome has been derived from Greek terminologies chroma and soma, where chroma means color and soma means body, which means that chromosome is a colored body. And it was named so because scientists observed that the special dyes stain the chromosome in a certain colored way. As a result of that, it was named chromosome. Now, what is a chromosome? Before knowing what is a chromosome, we should know what is a chromatin. When the eukaryotic cells are not dividing, the DNA and the histone proteins remain in a tangled mass of thin thread which is called chromatin, and they are of two types, that is euchromatin and heterochromatin. The euchromatin are the less dense region and the heterochromatin are the highly dense region. Now during cell division, this chromatin condenses to form highly compact structure which are called chromosomes. Now what is a chromosome made up of? A chromosome is a structure which is made up of a single strand of DNA and protein molecules which are histone proteins, and they are present inside the eukaryotic cell nucleus. Now a prokaryotic cell nucleus also contains a DNA molecule, but they are not present in form of chromosomes. In eukaryotic cells, during the cell division stages, the chromatin condenses as we said before, and there is formation of chromosome, where there is a supercoil formation of the DNA and the histone proteins. There is formation of nucleosome, and they are furthermore supercoiled to form a condensed structure which is known as chromosome. Now the main function of chromosome is to fit the long strand of DNA inside the nucleus of the cell. As we all know that DNA has got a large length which is pretty much impossible to fit. So to overcome that situation, the chromosomal structure is formed. Now if we look at the structure of a chromosome, it has got several parts. There is presence of a short arm which is indicated by P, and there is presence of a long arm which is indicated by Q, and between them there is presence of a kinetochore and a centromere, and the tail region of the chromosomes are known as telomere. This is the picture of a typical metaphase chromosome. Now these different parts have got different kind of functions, whereas the centromere is the genetic locus that specifies the site of kinetochore assembly and a site for sister chromatid attachment during the spindle formation, and the kinetochore specifies the attachment between the chromosomes and microtubules during the chromosomal segregation, and the chromatid allows to store two copies of their information in the preparation for cell division. Now the parental chromosome consists of only one chromatid, and after the duplication there is formation of sister chromatids, and the chromatids of the homologous pairs are known as non-sister chromatids. Now if we look at the stages of cell division, we can see that the chromosome undergoes to various changes in its structure. In the interphase, there is presence of a single chromatid in the parental chromosomes. After the DNA is copied, the chromosome pairs up in the prophase, and during the metaphase the chromosomes line up at the equator and there is formation of mitotic spindle. During the anaphase, the sister chromatids are pulled apart, and during the telophase and cytokinesis, the cell pinches in the middle and there is formation of two identical daughter cells which are diploid in nature. Now, if we look at the types of chromosome present in an eukaryotic organism, each of the cell consists of 24 pairs of chromosomes, and among them 22 pairs of chromosome are autosomes and one pair of chromosome is for sex chromosome. Now the autosomes stores many thousands of genes, and each of them performs a unique function for the development of the organism, and the sex chromosomes are present for the determination of the sex in the offspring generation. Now in case of male, the sex chromosome is XY, which is heteromorphic in nature, and in case of female, the sex chromosome is XX, which is homomorphic in nature. But there are few such exceptions where the number of chromosomes are not same, or they differ from the normal number of chromosomes present in an eukaryotic cell. Those are the red blood cells and the sex cells, where there is a difference in the number of chromosomes. Now, what is meant by a karyotype and idiogram? The number, size and shapes of the metaphase chromosome constitutes the karyotype or karyogram, which is distinctive for each species, and the karyotype have got several kind of characteristics, which are based on the chromosome size, the chromosome number, the sex chromosome present, the centromere position, the nucleolar organizer position, the heterochromatin pattern, the secondary construction and the binding patterns. Now, the karyotype consists of a photograph or diagram of all the metaphase chromosomes arranged in homologous pair according to the decreasing length and position of the centromere, which is described in the given picture. In the given picture, we can see the male and female idiogram of an eukaryotic organism. So hope this video will help you, and in the next video we will discuss about the variation in the chromosome numbers and what kind of disease and problems take place in humans due to these changes, and we will also discuss about the chromosome aberrations. And if you have any kind of query regarding the video, you can put a question in the comment box. Thank you.